Well, thank you very much to our panelists. We now have a mega panel of our previous speakers. Just to remind you, we heard about Prader-Willi, and we heard about genetics and some of the statistics of pharmacogenomics. We heard about an intriguing GWAS with the names changed to protect the innocent, and the Taylor PCI trial, and then you just heard our panelists. And so we now have time for about a little bit more than half an hour of discussion, and I think it's my job to start to instigate the trouble. And so let me just say that, building off of what Mark said, I used to think that this was an early drug development issue as well. You know, that we would have to determine these genetic biomarkers early in order to have any chance of making it in. But if we listened to our talks, which we did, we heard about a GWAS on, I think, a phase 3 or very late. We heard about redoing a lot of trials way after everybody in the practice community thinks that the question is either solved or never going to be solved. And so I want to ask the panel, what's up with the need for early versus late? And are these biomarkers, and is genetics going to be useful downstream more than upstream? Or is this just the selection of talks that we heard today?
So I think, Russ, first of all, that's a great question, and one I think many of us are, trying to answer. And so probably a little bit of both. Obviously, if you can understand a lot about your disease that you're addressing and the target, and if there's human genetic validation for that target very early, then you can begin to think about stratification strategies really early and build that in well. But then again, sometimes there are genetic variants that affect drug response, for example, adverse events, many you talked about, that you wouldn't necessarily be able to predict up front. So that doesn't mean you're going to catch everything. There's certainly pathways that aren't necessarily related to your target or even the disease that could associate with response.
Yeah, maybe it was Matt, you might have been the one who said if it's a hugely strong effect, it's your duty to find it early, but a lot of these stories are not huge. They're still clinically significant, but not hugely strong, and so you need the phase 3 or the bigger trials to see the signal.
Yeah, there aren't that many examples of things that have been found to date that you would actually find in a phase 2. But you can imagine what they might look like, and you would want to guard yourself to be able to do it. And I think that it can be done so efficiently. We essentially budget about somewhere between 1 and 2 FTE months of time for each PGX screen that we do for safety and efficacy. And that's, you're starting to get the price down to a point where, does it make sense not to be looking, even in some of these small phase 2 trials, even though there's only a very small pocket of possible effects that you could detect in that setting. The potential advantage of identifying it that early, I think, outweighs the costs.
I think we have some lines. What I'll do is I will bounce back and forth. That's just an arbitrary rule I made up.
Thank you. Khalid Bourin, Office of the Chief Scientist at the FDA. How many GY studies do we need to conduct in order to conclude that there is or there isn't an association with a drug response? Like the example that Matt gave in his presentation, do we need to have 2,000 study for the steroids response in order to conclude that there's no association?
Well, what you can do is you can, based on the statistical power you have in any particular study, you can describe the space of effects that you can conclude do not exist for a particular drug. So you can essentially say, based on the statistical power we have in this, we can be fairly confident. You can even take it from a Bayesian perspective and say, we can have 95% confidence that no effect of this size and of these frequencies exist for this drug within this population. So you can start that even with very small studies. You can start to essentially say, okay, well, there's nothing out there with an odds ratio of 100 and a frequency of 70%. That exists that could be affecting the response to this drug. And as you get larger and larger studies, at what point do you reach that you say, okay, we can rule out that there's anything that could possibly be clinically significant for this particular drug?
So it's case by case study.
Well, how quickly you can accrue the samples and the evidence to be able to kind of reach that level of confidence that there's nothing left that would be valuable to discover is going to be case by case. So some of those may be, within the context of clinical development, and some maybe 10 years after, when you can finally collect that amount of data.
Bob Wilden, NHGRI. I'm going to say a couple perhaps provocative things. The first thing, so I've been driving for 40 years, and I have never been in an auto accident where a seatbelt would have saved my life. Does that mean I should stop? That I should stop wearing seatbelt? I don't think so, especially not in the DMV area. Okay, so the risk to me is relatively low, but the consequence to me or to whoever it is who gets in a wreck with a seatbelt on or off is relatively high. So I want to start maybe a discussion about this concept of the predictive value being too low when the odds ratio is actually quite high. And what that means, and this is particularly important also for rare disorders, is that the person who is affected by it is left out. So the question really shouldn't be what's the predictive value, but what do you lose by making the recommended changes in dosage or choice of drug? Does that have any harmful effects on that group that is at risk? And if it doesn't, then you should do it anyway.
Well, since they all punted to me initially, or at least Matt did initially.
So clearly there are economic reasons, but putting the economic reasons aside, and I'll point out that if everybody had a genome on file, there would be no other economic argument.
Well, but it goes beyond just the economics in terms of what the cost is. You have to get into competing risks and lots of other factors to decide whether it's useful at all.
Yeah. And a lot of what I took from your example was the effect size, right? Your effect size was death versus minor inconvenience of a seatbelt. And I think in drug development, we see a whole range of effect sizes of both data collection itself isn't cheap and can be invasive and cause its own risks to patients, right? To some of the effect sizes appear to be pretty small. And if you're looking for, if the seatbelt only avoided mild bruising as opposed to death, I might have a different view, right, on all these.
So what's really challenging is it's kind of easy to say, okay, well, we could reduce the risk of developing this particular adverse event by twofold. If we magically had all of that genetic information readily available, the question then is, but then what are you going to treat them with if you're not going to treat them with drug A because they have a particular genotype? So then you move to drug B. Well, what are the risks around drug B? What are maybe some of the genotype-specific risks around drug B? And so you essentially have to put all of this information together into a single model to be able to make a rational decision about what essentially maximizes that benefit risk.
I think Scott's trying to get a word.
Scott?
Go ahead.
Adding on to this, taking a real perspective, comprehensive approach de-risks some of these issues. I think we heard a little bit today already that when you try to go back and do these analyses, it becomes a real challenge. But when we plan ahead and make the collection of the genomic information and actually set up the infrastructure, the systems, and the roadmap to collect this info, it becomes much easier to do prospectively. And then you've also set it up to take that retrospective approach when you need to. And this is a very provocative comment, but couldn't genomic profiling become another vital sign at some point? This is really a new era of technology to be able to do the kind of processing of this information, and now the ability to apply this to actually affect care. We're definitely in a different period now.
I've worked in clinics where getting an accurate blood pressure is about as expensive as a genome will be very soon because of the number of bad blood pressures that we measure before we actually get a good one and then act on it.
But can I just say, I would argue, though, that I think we do not have anywhere near enough evidence to be able to make good decisions about what alternatives there are. So we can talk about, what is the advantage of this drug for this patient, but you have to count, okay, so if not this drug, then which drug, and what are the benefit risks that go along with that? And I think that there are very few circumstances where we have the kind of evidence accrued where we can actually make a rational decision.
I think you're right. We don't have it now, but this is building for the future, using the perspective approach to set us up with the new tools and emerging technologies to start to make those decisions.
I'd like to make a comment to the earlier question about how many GWAS studies are needed to prove that a study or a drug doesn't have a strong genetic factor. I think that it's important to recall that a lot of the GWAS studies that have been done today are in one population group, that's in Europeans or European Americans. And so we're not even targeting the populations that are oftentimes at highest risk for the disease outcomes. When you look at asthma rates in terms of death, hospitalizations, ER visits are about 3 times higher in African Americans. When you look at diabetes, rates of microvascular disease, macrovascular disease are much higher in African Americans. And yet, in the GWAS studies that have been done for it, metformin, the drug of choice for the treatment of diabetes, it's almost exclusively in European Americans. All the studies that have been pointed out that have been done for inhaled corticosteroids, again, almost all in European Americans. So if you're not targeting risk groups, groups that we've shown benefit differently from drugs or are at highest risk for outcomes, and we really can't say that the GWAS studies have been sufficiently covered in terms to say that there's not a genetic risk overall, and that we can't identify biomarkers that are important in populations.
We're looking for our keys where the light post is shining, perhaps.
Hi, Bray Patrick Lake, Duke University, but I am a patient person, and that is my primary lens and what I bring to this work. And every single comment that I've just heard plays into something I've been trying to wrap 2 brain cells around to say, we hear that, you do this GWAS, we don't know who's at risk. So what solves that? Volume. What solves that? Data quality, connections. We've heard from all of you early adopters and innovators, these great projects that you've done, but they've been done in isolation. We've got to have one system where we can all plug in and be part of this. And it's not a continuum, it's not linear, it is a cycle. So my personal patient story, I actually had a major clopidogrel accident. My ALT was 470. Do I look like somebody that would be even in the trials that are being designed? Nope. This information is completely not being collected. So, if we're really going to get smart about identifying new targets, expanding labels, identifying adverse events, using real-world evidence, we're going to have to, get past this. I've been to I don't know how many meetings about, what we need to do as data sharing and creating a system. But hopefully coming out of this, and if we keep I don't want to go to another one of these meetings. They're great, but I really don't. I want us to actually take action and do it. And so maybe today we'll all connect and we'll leave, and instead of being rare diseases and chronic diseases and complex diseases, we'll just say that we're going to create a system for all patients. We're going to collect all data because there's the uncertainty. We don't know what we're going to learn. And I also want to say that patients can understand and handle uncertainty. It's the Jack Nicholson line of, you can't handle the truth. Well, the truth is we can. It's just explaining it to us. And so I think it's fine if we all become a little more comfortable with risk. But I do want to say we have to stop these one-off projects, even if they're fabulous. We have to stop and all come together.
I would agree. I mean, I think that we as we've shown, we have a way of not only collecting data on everybody, but we have ways of identifying populations that are at risk. And we should be focusing in terms of identifying biomarkers in groups that are at highest risk.
Yeah, I think everybody would probably agree with that statement. I think the challenge is how do we do it in this society that's, first of all, capitalistic here in America and very used to having competing, whether it's products or methods or, that's just a part of how life is for us. So how do we don't have patient registries really in a centralized way like some European countries do. We don't have a single healthcare system. So, how do we do that, I think, is the problem. But I think everybody, including pharma companies, by the way, are interested in participating in an integrated and pre-competitive way to conduct genetic research in large numbers of people.
Yeah, just adding to that, I think what we see here is how important the partnership really is, the academic, industry, vendor, patient advocacy group, and health authority, you know, this multifaceted partnership that really enables this kind of connection with a common, like you said, pre-competitive approach, and maybe up to all of us to drive this.
Hi, I'm Mike Doherty. I'm the Director of Education for the American Society of Human Genetics. We've heard a lot today about the challenge of recruiting sufficient participants into clinical trials to reach outcomes that really help inform the integration of drugs effectively into clinical care. I'm wondering, there are lots of folks on the panel up here who are involved in putting together big clinical trials. Are there systematic best practices for education of providers and participants that you're employing to try and improve the recruitment outcomes?
Best practices for
sorry.
recruitment. Oh, I
I'll
was just killing time.
Yeah, sorry, I'm unable to work the microphone, so I'll start. So when you ask people where they want to hear about a clinical trial, most say they want to hear about it from their primary care physician, yet nearly the overwhelming majority of primary care physicians know nothing about clinical trials. They wouldn't even know where to send their participants to. So we end up with only specialized medical centers, specialized practices that are offering them. So it really is how do you even bring awareness to the primary care doctors? And they can't be aware of every single clinical trial that's out there for every, look at the breadth of disease areas that we're talking about today. So even the thought of having sort of having a good go-to national website of sorts, clinicaltrials.gov doesn't serve the purpose of being friendly with lay accessible information. That's supposed to be the resource of where every trial is and every research study, but you go on it, you read it, and you're immediately lost. And so we need to really start to drill down of like, what are the salient pieces of information that people need to know about a study, and how do they take the next step should they be interested in doing them? We take a lot of time in just educating people about what it means to participate in a clinical trial. What are some of the things that you might be asked? Why is it important to sort of demystify the process so that when they are invited to a study, they come prepared. We created educational videos about considerations for learning your ApoE genotype to participate in a study. And I can tell you, when then people come in and they sign consent and then meet with a genetic counselor to learn, they are very well informed. But it's putting all of that upfront energy and effort into place to creating these educational materials and not leaving it to the study coordinator at the site who is, overwhelmed and trying to educate a bunch of participants about a range of studies.
We didn't get a lot of other people jumping in.
So,
Let me ask if it would be helpful if, rather than doing it on a one-off basis, every clinical trial goes about developing its own education materials, would it be helpful for a national group like ASHG, or perhaps ASHG working with others, to develop some generalizable education materials that could be a go-to source for providers, especially primary care, where, as you say, patients are interested in getting the information?
I'll say yes. So I think from the rare disease standpoint, the different advocacy groups have different levels of sophistication on the same thing. And when clinical trials come into a rare disease, the patient base may or may not be sort of educated and understand what to be asking. And so a resource from ASHG or would be really useful that, advocacy groups that maybe, don't have a geneticist on staff, can get the information and share with their families. So I think there's two things. There is educating the providers, the primary care physicians, but I also think it's educating and empowering patients themselves, and especially those that are already identified with, most of the rare diseases don't currently have a treatment. Hopefully there'll be lots of clinical trials moving forward, and so getting everybody on board now about what a clinical trial is, I think would be really help facilitate the whole process.
and coming at it from a different angle, from the non-rare disease side, University of Pennsylvania and NHS in England, UK, have also shown with their level of databases today, and they took about a half a dozen different clinical trial protocols with all the inclusion and exclusion criteria, lab values, et cetera, age group. They could identify enough patients in each of those systems individually, and for the UK it was just like one metropolitan area of 10 general practice to fill that clinical trial given normal recruitment rates. So they had enough patients that they could identify that met the criteria that if only 1 out of 20 participated in the trial was filled in 6 NHS sites in the UK or in the University of Pennsylvania system. Now, the Pennsylvania system, as you might imagine, was much better at hospital-based clinical trial protocols, not the outpatient side, because that was their data system strength. NHS was pretty powerful on both and was really powerful for some asthma studies, quite frankly. So there is a different way to come at this, which then if we had those kind of materials, you might have more than.
I can't tell if this whole line is all for questions, but it doesn't.
So, yeah, I have a comment and a question. My comment really echoes Bray's point about everybody working together. And as I look across this panel, it's really interesting to see two pharmaceutical companies really struggling with the economic case, to the case and the business case to really fully integrate genomics into drug development. Yet at the same time, we have foundations and patient advocacy groups that are gathering patients, genotyping them, and channeling them into clinical trials. So it seems like there's a natural marriage, as has been said, between the registry groups and the drug development firms to really put these resources together in a way that maybe hasn't been done before. Several years ago, we had a workshop here that led to a framework for pre-competitive collaboration for genomic-based discovery in drug development. Not quite the topic of today, but what it ended up doing was getting a number of companies to pool their control arms from clinical trials that had genetic information and actually use that as a way to do discovery. So I urge that we might consider revisiting that framework as part of this workshop. My question is really for Michael, and I'm just dying to know what compelled the Mayo Clinic to invest $3 million in a clinical trial on clopidogrel, because I really would love to understand the incentives behind that, in light of some of our discussion this morning.
Yeah, I mean, they had developed a Center for Individualized Medicine, and they had focused on key areas, cancer, cardiovascular, and they went to the cardiovascular community and said, what question is burning in your practice? And they came up with this. I was shocked by that too, to be honest with you.
But what's the return on that investment that they saw up front? Did they see, did they do modeling to show that if the trial were successful and then future patients were genotyped, that this would obviously save lives and save money? I mean, was there a very cogent, compelling argument along those lines?
I think what happened is Mayo invested in a database adverse effects of drugs and devices unit and the SIM, and they wanted to do things together. And I think this would exactly what they thought of this a question that could have an impact across more than one unit.
Yeah, it would just seem like some of that thinking, whatever it was, would really play a role in a lot of our discussion today. And Mark, you may have seen some of these types of models as well. But I think that's in designing these studies, as Merck and GSK have to do. We have to understand what's going to happen at the other side of the trial if it's successful.
In the same way we were just discussing templates for patient participation, we should have templates for how to convince my hospital administration at Stanford to make huge investments in clinical trials like this. I mean, it's only a little bit of a joke. There is, I agree, a certain appeal there, and that case needs to be made across the country, really.
Yeah, my personal experience has been a strong-willed philanthropist is usually the catalyst.
Yeah.
Yeah, it was in the Mayo development of the unit. It was a breast cancer patient, wealthy family that gave the money for the unit, which then led them to expand it to cardiovascular.
So it goes back to patient recruitment. I think I'm on this side now, and then I'll do this side.
So I think building on Jeff's question, Mike Pakinowski, FDA, $3 million could buy 3,000 whole genomes, which I think would be one way to spend that money. But in the common disease space, we have to think about things like population attributable risk and numbers needed to screen, at least in the sort of current environment. A hazard ratio of 0.65 is a huge effect by cardiovascular standards. So, considering what ticagrelor and the newer drugs do relative to clopidogrel, sort of at the overall population level in terms of an effect size, number one, do you think that's an adequate effect size to shoot for, and will you realistically meet that? And then two, do you consider some of these population attributable risks and other factors into determining what would be a relevant effect size to find in this type of trial?
Yeah, there was multiple meetings of the investigators, and one of the things is the absolute risk reductions that we would see. And one was convincing people that an individualized approach would really change practice. And that's where they came to the threshold of 35. But some have even argued for 50% reduction. If we don't show 50, then we really haven't been convincing enough to change the practice. Because obviously, if this trial is positive, we would advocate that everyone be screened who's on clopidogrel, and that could be a lot of patients. So the issue was it came down to, when we do non-inferiority trials, we define the margins that are important, and it's done by committee, and that's how they came up with this 35%. But they lowered it to 35 because the NIH wouldn't fund it if it wasn't as low as 35.
Okay, thanks.
Hi, Melissa Clark, Howard University. So I wanted to get back to the asthma question. So asthma is sort of like the textbook disease for social determinants of health. And, because it, is impacted by environment and education and access to care, transportation, housing, all those various issues. And so as you look at ethnic differences in, I think the slide that you showed, you mentioned the left shift. As you look at those ethnic differences in compliance with medication, how much did you sort of control for socioeconomic status? And the other reason as it pertains to genomics is also looking at some of the chronic stress that comes along with being on the negative end of the social determinants of health, and how those epigenetic factors may also play into frequency of exacerbation, and therefore less of a drug effect, or even maybe epigenetic factors that may affect drug effect.
Sure, great question. So we've looked at the issue of the factors related to non-adherence, both existing behavioral models, a number of existing behavioral models of different metrics of disease perception as well as belief in medications. We've looked at some of the social and socioeconomic determinants. And even after adjusting for most of those, we still see a persistent difference in utilization. And there may be obviously unmeasured factors that seem to be determining the differences in use that we see. One of the papers that we wrote actually was looking at crime rates in neighborhoods as a factor for determining medication adherence, and we did see a relationship with crime rates in the city of Detroit. So your points are very valid, and thinking outside of the box, I mean, there are probably a number of different reasons that we see differences in utilization. We have looked at differences in response rates for inhaled corticosteroids, and fortunately the response rates when we adjust for use appear to be similar, so it doesn't seem to be a difference in treatment efficacy for inhaled corticosteroids. So there are probably a lot of different determinants that go into use. Some may be response, some may be side effects, some may be the social determinants that you were speaking of. But we haven't found, it's a complex issue, and we haven't found one factor or a group of factors that's explained all of that difference yet.
Hi, Bray Patrick Lake again. So some of the bigger issues that we're continuing to explore that made me think maybe we don't have all the stakeholders in the room. Do we have health systems here today? I know we have people that work for health systems. Do we have CEOs, leaders of health systems? And do we have our EHR vendors and our our informaticists in this room. And I bring these points up because we're continuing to talk about research over here and care over here. And wouldn't it be great if providers refer to research? Wouldn't it be great if we had diversity in clinical trials? But we actually have diversity getting care on a regular basis. We also have opportunities to learn from people and build things into the EHR on a regular basis. And Dr. Falk, was talking about, we asked patients what was important, and look, lo and behold, those turned into PROs and they turned into clinical endpoints. We actually use them in clinical trial applications, but again, we're going over here to collect information that we could be collecting and gathering every day. Then when we identify, a new mutation, something that might be promising, you don't have to go back and do all this foundational work. So again, I just want us to start thinking more about the larger opportunities if we're all one system.
Great point. I think there is a precision medicine initiative that actually is recruiting from health systems, including Henry Ford Health System. So, I mean, that's people are looking at the ways of utilizing the EMR and leveraging that in the setting of precision medicine, and hopefully those data will be, it's an ambitious project to recruit that many patients representing such a diverse swath of the U.S., but it's ongoing, and hopefully once that is completed, we'll be able to address some of the issues that you're talking about.
Yeah, again, so I work closely on that intimately.
Yeah.
All of us research this program now, but we're talking about recruiting people to be in. They shouldn't have to try to get in. They should already be in when we get to the larger system.
Right.
We have about 3 minutes before our break, so I've declared this now the lightning round, where we will have, I don't know what that means, but please try to behave as if it were lightning.
Okay, here goes lightning. John Cabral from FDA. We'd like to highlight a couple of things and then pose a question. First, I was in this room about almost 2 years ago when we convened a meeting on recruiting and diversifying clinical trials. And at that time, there's a wealth of literature, strategies, and it's really disturbing just to see that we've got a cardiovascular trial underway with 2% Blacks in it, which takes me to two other initiatives I've had the opportunity to be part of. One is the Clinical Trials Transformation Initiative, which I was part of the recruitment group. And one of the take-homes from that is that there needs to be a plan looking at who are the appropriate patients, the clinically relevant patients, at the beginning, that it's not an afterthought where you have this aha moment, my goodness, we don't have a representative sample. The second is that at FDA, we issued a guidance document, an update actually, that provided more explicit direction to commercial sponsors on having the clinically relevant patients in the trials and to plan for it no later than the end of phase 2 meeting, but to include that as part of your development program. Because if there's anything, and I'll echo Ray's earlier frustration, is that it's really tiring to continue to see data that doesn't reflect the people you want to have a positive outcome on at the end of this process. And so I also put this question to the folks from industry who are doing the genetic studies as to how are you incorporating this into your planning. So thank you.
Some lightning answers, please.
Cardiovascular trial. Your points are well taken, and in fact, when we got NIH funding, the sites that we're adding, the additional 12 American sites, are all in urban and centers where we can recruit, because as you know, NIH has made that a very, very big policy about minority recruitment and those kinds of communities. So we couldn't agree with you more. I mean, to get the trial off the ground for the prices we were paying, we weren't able to go to those centers to create the infrastructure. So we're on track with you there.
And, Russ, one of the, so for the discovery efforts that we're conducting that Matt and I talked about, we're kind of beholden to whatever is enrolled in the clinical trials, right? And I think we all know that they aren't terribly diverse. However, once we have a finding and we want to replicate that finding and we understand what the allele frequencies are by ethnicity, then we can target a population. And I think many of us are doing that, right? So we have one trial ongoing right now in Asia-Pacific countries, specifically because we're testing a hypothesis for an allele that is prevalent mostly in Asians. So I think we're going to see more and more of that once we start understanding by ethnic distribution of alleles, who we should be addressing.
And just one quick comment to add, and as we are identifying new therapeutic opportunities that may be defined by particular alleles in the population, I think that is driving us to different regions, different geographic subgroups, to be able to treat in those ways. And so I think we will find in the future some of those underserved populations as the specific target for new development opportunities.
I think there's an underlying fallacy here that people aren't recognizing, and that is if you actually do your studies primarily in European Americans and you discover an allele that's prevalent in one population and not prevalent in another, you can't use that as evidence that that's the reason that there's a difference in response between the two population groups. If you actually look at the GWAS data, they're actually finding different alleles that are risk factors for disease. So why would we actually assume that if we were looking solely at allele frequencies, that's going to explain the difference in disease for these pharmacogenomic predictors? It may be that there's completely different alleles that are risk factors in different populations. And so if you don't primarily and adequately power your studies to look at those populations individually, you're not going to be potentially identifying the important relevant risk alleles.
We have one last lightning comment.
So asthma mortality and morbidity is fourfold higher in African Americans and Puerto Ricans, and the GSK study only included 5% African Americans, and so nearly effectively closing the door on inhaled corticosteroids, which are the main controller medication for asthma in in all children, actually. So I'll leave it there.
Okay, well, with that, I want to thank the speakers and the panelists for a great session.